Good evening. Thank you. That was a very kind introduction, probably the longest introduction I've ever received for a talk. And so I would really thank you for the invitation to be here. It's a beautiful city. The little bit I've seen, I hope to see a little bit more tomorrow. It's always wonderful to come to Hungary. And I truly have always enjoyed my interactions with people here. I would like to start my presentation with a couple of apologies. I didn't quite know what audience to expect, and so the talk that I put together is a scientific research talk. And I realized that I should probably have rather put together something a little more historical and more explanatory for the students than I have done. So I would apologize for that. The other thing I want to apologize for is that I put together a talk that was timed to be for 60 minutes, and it's 7 o'clock, and there's dinner. And there's no way you would allow me to talk for 60 minutes, okay? I realize that, okay? So I'm going to skip over some parts and hope that that won't ruin everything, but I think that that would be a wise decision. And I will try to fill the gaps as best as I can, but if there's any questions or anything, please don't hesitate to ask. And finally, I'm going to talk about stuff I'm interested in now. I'm not going to talk about neurotransmitter release or vesicle traffic or any of this. I still think that that's a very interesting topic, and I am always delighted to listen to it, to hear from others, especially my former co-workers. But right now, I want to address a totally different question that has really fascinated me for a number of years now, and that regards neurotransmitter release. That regards synapse formation, not neurotransmitter release. You can see I'm jet-lagged. Okay, sorry about that. By way of introduction, since I'm the neuroscientist among the group here, I think you all realize that the brain is rather complex. It's huge. There's trillions of neurons connected into vast networks by thousands of synapses each. The brain is a lot more complicated than the human genome. We don't understand the human genome. We all need to work on it because in the end it is what dictates biology. But the way how it does it is incredibly indirect and complicated. If the human genome is so complicated, you can just anticipate how complicated the human brain will be. And so it's not surprising that neuroscientists, that are a rather diverse bunch of people, have little to agree on. There is in the neuroscience community a huge amount of discussion, arguments, sometimes not very friendly, but in general, there's this concept of neural circuits that I think all neuroscientists agree on, and that it forms sort of a kind of almost mythical basis for neuroscience because it's actually not a good concept, but it's useful, like so many concepts. And that concept of neural circuits is illustrated schematically here for a microcircuit. That concept says that synapses connect nerve cells into communicating networks, circuits, and information is processed as it flows through that network. In the circuit, the synapses connect the nerve cells. They transfer information from one nerve cell to the next, and they change that information. They process that information. The synapses are the fundamental information processing unit of the brain, because synapses are not transfer station of a signal from one nerve cell to the next. Instead, synapses differ very, very much among each other depending on the identity of the pre- and postsynaptic neuron in changing that signal as they transfer it. And this is unpredictable. You actually have to measure this. As a result, if you look at a simple circuit as this one here, where you have a presynaptic neuron that excites both an inhibitory and an excitatory neuron, and the inhibitory neuron then has a feed-forward inhibition on this excitatory neuron, the relationship of excitation here to a signal there cannot be predicted, because the synapses that connect these various neurons could have different properties. They could be facilitating or depressing. They could be all kinds. Okay? And as I said, you can't predict this. Moreover, to make things complicated, there is various parameters of synapses that make them different. They have different forms of plasticity, different receptors, and so on. So circuits, as a basis of information processing, are in themselves not simple. Now, just imagine that the brain is composed of millions and millions of circuits that are interdigitated, that are overlapping, that are parallel, that are sometimes local or can explain the entire brain. You can, I think, get an idea of why the brain is so complicated. The question that I have been interested in for the last decade or so is, how are synapses actually made and how are they specified? A problem of neural circuits, one of many of understanding neural circuits, is that neural circuits are not hardwired. Although these synapses are made during development, they don't just stay there. They continuously change. They are eliminated. They are reformed. In some brain regions, like the hippocampus, a synapse has a half-life of 2 or 3 weeks. So it's not easy to think of the brain like a computer, because different from a computer, the brain is sort of a computer that constantly rewires itself, as opposed to a computer which has a hardware that is then changed by software, but in itself doesn't change. What I am interested in is try to understand how synapses are made, not only during development, but afterwards, how they are eliminated, how specificity is achieved, how plasticity is achieved. In order to pursue this question, we posit that synapses, as shown here in this electron micrographs, are intercellular junctions, that they are connections between two cells, two neurons, and that as intercellular junctions, they're connected by molecules, so-called adhesion molecules, that span the cleft between the two cells. Adhesion molecules are called adhesion molecules because the idea was that they would make cells adhere. But their real function is not adhesion. Their real function is signaling. They signal, and the idea here is that these adhesion molecules that govern synapses, synapse formation, synapse properties, and so on, are signaling devices. That they signal bidirectionally and thereby allow these two neurons that are connected at the synapse to talk to each other, to tell each other how to behave, which is also going to be the subject of my talk today. Not surprisingly, given the importance of this subject, many synaptic adhesion molecules have been described. This shows schematically a selection that I think might be relevant at some point. And you can see that on the left side are the presynaptic adhesion molecules, on the right side the postsynaptic ones. There's more postsynaptic adhesion than presynaptic adhesion molecules. Many of the presynaptic adhesion molecules are hub molecules like neurexins or PTPRs that interact with a lot of postsynaptic molecules. The description of these molecules is a bit confusing because there's so many of them. And like so often, when in science we find something, it is difficult to tell from the beginning what's actually important. It takes a lot of time. Very often you stumble upon something and think it's really important, everybody else thinks it's important, it turns out to be totally irrelevant. And conversely, sometimes you have to have the conviction to pursue something with a gut feeling, even though nobody else thinks it's important. We are trying to figure out which ones of these molecules are important and what they actually do. It's clear that synapses are not a system where you just have one set of molecules, a few molecules, and that they basically instruct a synapse. No, a synapse is going to be constructed by the combinatorial code of many molecules that act together. There's not going to be one master molecule, although it turns out that there's a few molecules that are a bit more important than others, as I will describe to you. I'm going to focus on one set of molecules which are called neurexins, and which are these molecules here, because they are probably the most important presynaptic hub molecules that do a lot of things at the synapse by directly or indirectly interacting with the majority of postsynaptic adhesion molecules. So they're kind of key regulators of the process. So when I initially planned this presentation in the assumption that this would be a regular science talk, I had planned to talk about all this stuff, which I can assure you now I won't, because, as I said before, dinner is waiting. Instead, I would like to propose that I'll just give you an introduction to neurexins as a class of molecules, and then tell you just about one particular feature in my presentation, namely the regulation transsynaptic control at a subset of synapses, not all synapses, just a subset of synapses, of postsynaptic receptor responses by presynaptic neurexins. So you can get a feeling for how this works, how we might be able to put this together, so that you can understand why I talk about the molecular logic of the coding of synapses that underlies the specification of their properties, and so that you can also get an preliminary insight about the kind of approaches that we use to eventually get there. It's going to take a while, though. And I hope that that will give you some kind of impression of where this field is going and how we might, in the end, be able to understand all of this. So let me introduce the neurexins. What you see here schematically is a synapse with a bit larger synaptic cleft than it would normally be. And neurexins are shown on the left side. There's two principal forms of neurexins called alpha and beta neurexins, of which the beta neurexins are simply shorter versions of the alpha neurexins that are produced by an internal promoter of neurexin genes. So alpha neurexins are composed of these domains called LNS domains. This is what the L stands for, and EGF-like repeats. That's what the E stands for. So these are independently folding domains like pearls on a string that are lined up here. There's 6 LNS domains and 3 EGF-like repeats. The beta neurexins only have a single LNS domain. And these are interaction domains that are found in many proteins. There are type 1 membrane proteins, as you can see here, and there's also schematically shown here they interact with a number of postsynaptic ligands. There are 3 genes for neurexins with independent alpha and beta promoters. So there's 6 principal forms. In addition, the neurexin 1 gene has a 3rd promoter for a gamma neurexin that I'm not going to discuss. Neurexins, as you can already assume from what I said, are localized to synapses. The way how we show this is by immunocytochemistry. What you see here are confocal images of synapses in a cultured neuron that have been stained for a neurexin 1, that's actually epitope tagged by knock-ins. It's the endogenous neurexin, and either presynaptic or postsynaptic markers. And you can see that there's pretty good co-localization. Now, this doesn't tell you where exactly in a synapse a molecule is, because synapses, although they look small, are actually quite big. It can be a micron or so. And the resolution of confocal microscopy doesn't allow you to find out where exactly it is. For example, you couldn't tell from these pictures whether neurexins are pre or postsynaptic. To achieve that, we collaborated with Xiaowei Zhuang at Harvard University some time ago and performed super-resolution microscopy. This shows a number of images obtained either in cultured neurons or in tissue sections, hippocampal sections, that illustrates that the green neurexins compared to the red junction, the red synaptic junction that is labeled here with HOMA1, which is a postsynaptic marker, only occupies a tiny bit of the junction and forms a nanocluster. This can be quantified. Shown here are the nanoclusters. This is a bigger magnification. There's about 5 neurexin antibodies per cluster, and the size of these clusters are similar in cultured neurons and in tissue sections. Since then, a large number of other synaptic adhesion molecules, including several neurexin ligands, were also shown to be localized to nanoclusters, both using D-storm super-resolution and STETRO super-resolution, approaches that have really changed how we look at things, because they allow us, if not nanometers resolution, at least tens of nanometer resolution, which is pretty good. So you have these molecules at the synapse. And obviously the question arises, why should you be interested? Now it turns out that neurexins are not only conserved hub molecules at synapses, but they are also relevant for disease. Thousands of patients have been identified. So this is not super rare, that contain heterozygous deletions, in particular in neurexin 1, but also 2 and 3. These patients were identified because they had neuropsychiatric disorders, but not just one. They either had autism or schizophrenia or Tourette or intellectual disability or a combination thereof. So there was no specificity. When you look at where these deletions are within the neurexin gene, you can appreciate from this slide that maps the subset of these deletions that they basically cover the entire neurexin gene. Alpha and beta. Beta is here. This goes in this direction. There are so many deletions because it turns out that the neurexin 1 and 3 genes are huge. They are more than a megabase. They're enormous. They're one of the bigger genes in the genome. We're full of repetitive elements. There's nothing else in there. There's no non-coding DNA, RNA. There's nothing. And all of these deletions are basically loss of function deletions. Surprisingly and puzzlingly, when patients were analyzed, it was found that there's no correlation with the actual deletion and the clinical presentation. Sometimes in families, siblings with the same deletion would have completely different clinical presentations. This was puzzling and raised the question whether these deletions are indeed actually pathophysiologically relevant. In other words, if you have all these patients with these deletions, maybe it's an accident. Maybe this is just a very sort of vulnerable gene that often gets mutated, and the accidentally is present in these patients. And so the question arose whether this kinds of heterozygous deletions has any effect whatsoever biologically. And that question was amplified by the observation that, at least in our lab, the heterozygous deletions in mice have no effect that we could measure. So to address this question, we pursued a number of approaches that are based on technology that we developed over the years, which enables us to rapidly convert stem cells into neurons, a technology that is based on forced expression of NGN2 as transcription factor, and that has been widely adopted by, I think, hundreds of labs by now because it's so convenient. And we use that technology to either convert engineered conditional heterozygous neurexin-1 mutant stem cells or patient-derived stem cells, induced stem cells, iPS cells, into neurons and to analyze them. The neurexin mutation deletion had no effect on the conversion of stem cells into neurons. The neurons looked just fine, as you can see here in this slide, but as may be expected, with the heterozygous deletion, there was a decrease in expression, not quite 50%, about 40%. So there's some compensation from the existing allele, but showing that the gene dosage is actually rate limiting for the expression levels, which is not always the case. Again, one of the things you can't predict in the human genome. Neuronal development was normal. Synapse numbers, we couldn't find a change. So we looked at it functionally using electrophysiology, and we found that in these human neurons, the deletion of neurexin-1 heterozygous caused a significant decrease in synaptic connectivity and synaptic strength, as shown here with these evoked responses. This was a discrete phenotype that likely reflects an impairment of a specific step, namely, interestingly, neurotransmitter release, in fact, in synaptic transmission. So when we did this and we published this, the concern was that the conditional engineered mutation that we performed that was designed to control for genetic background was a very artificial way of doing this, and people wanted to know whether it was actually a reproducible finding and whether it could also be seen in neurons derived from human patients with neurexin-1 deletions who had a clinical presentation, in this case of schizophrenia. So we started a large reproducibility project that used 3 pairs of patient-derived stem cells coupled to stem cells derived from normal controls, unrelated, of course, no control for genetic background, unfortunately. And we used these stem cells in our labs at Stanford as well as in the lab for completely independent investigator at Rutgers in the United States to make neurons and to compare their functional properties. I'm only going to show you one slide of this. Again, neuronal development was normal, synapse numbers were normal. There was no change in that regard. But as you can see here in these 3 pairs, shown in different colors, in each pair, relative to the control, there was a decrease in synaptic strength and an increase, what's called the coefficient of variation, which is an indirect measure of the release probability and measures an increase means there's a decreased release probability. This was confirmed with paired pulse measurements. Not going to go into this. Trust me, it means that there's a change in short-term synaptic plasticity, which basically means, in this case, most likely a decrease in release probability, and this was independently confirmed at Rutgers, as shown down here, with slightly different approaches, same conclusion. So these experiments thus confirmed that the patient-derived stem cells also had a major phenotype, suggesting that indeed, actually I think quite documenting, demonstrating that the heterozygous deletion of neurexin 1 is biologically significant, that it actually does change the neurons, at least those neurons that we make here in a dish. We confirmed this further with other approaches. We also confirmed by doing exactly the same experiments for mouse neurons that you don't see this in mouse neurons. So for whatever reason, mouse neurons are not as sensitive to the gene dosage effect as human neurons, demonstrating, among others, that when we look at disease models, it's always important to approach that from as many avenues as possible at any given time. Finally, in my introduction to neurexins, let me emphasize a very important point, which is that neurexins are alternatively spliced at 6 canonical sites, which creates thousands of isoforms. These are highly polymorphic molecules on the surface of neurons that are expressed in a vast number of combinations. This was confirmed using PAC biosequencing, for those of you who are aficionados, which enables us to actually show for each individual mRNA molecule what splice variants are actually present, and demonstrating that this is indeed a very, very polymorphic molecule. Moreover, these splice variants are expressed in a highly regulated manner and in patterns that are specific for particular types of neurons. So presumably, they should be meaningful. So what I'm going to do now is tell you about a key function of neurexins in this bullet point 2, and then I'm going to skip the next 2 bullet points because I'm assuming at least you're hungry. If you're not, you're going to have to eat nevertheless. So in this project that I'm going to describe to you, I'm only going to talk about one particular, as it turns out actually, restricted facet of neurexin functions. They do many other things. But I'm going to tell you about this because it revealed a specific function for alternative splicing of neurexins, a function that is very important for understanding synapses. It's been known since the pioneering work of Petersen, who, as you all know, is one of the really most impressive neuroscientists, Hungarian neuroscientists, in the last 50 years, that synapses contain different amounts of receptor molecules, and they contain different combinations of receptor molecules. In excitatory synapses, glutamate is the neurotransmitter, and there are at least 2 principal forms of postsynaptic receptors called AMPA and NMDA receptors. And the relative mix of these receptors is characteristically different between different synapses. There's also a 3rd form of receptors called kainate receptors, which I'm not going to discuss today. The results of our study on SS4 unexpectedly addressed a key question in neuroscience, namely the question of how the content of postsynaptic neurotransmitter receptor in a synapse is controlled. It didn't answer that question for all synapses. There's more mechanisms that I don't have time to talk about and that actually we recently discovered. And so this is really just gives you a glimpse of it. Nevertheless, for those synapses at which this mechanism operates, it's profound. So how did we approach this question? Alternative splicing of neurexins is obviously encoded in the genome. In the genome, splice site number 4 involves an alternatively spliced exon, which in the case of neurexin 3 is exon 20. This exon has an unusual splice acceptor sequence that is non-canonical. What we did is manipulate the mouse genome to convert this unusual splice acceptor sequence into a canonical splice acceptor sequence, which rendered this exon no longer alternatively spliced. With that mutation, alternative splicing no longer operated, and this exon was always included. So it would be always SS4 plus included. However, we also flanked this with LOXP sites. So when we take Cre recombinase, we take it out, and it becomes constitutively excluded. In other words, without Cre or with a control, which we call delta Cre, which is basically a Cre recombinase that is mutant and inactive, but otherwise the same, there's constitutive inclusion of SS4. With Cre, there's constitutive excision of SS4. We made these mice. We started with neurexin 3, and we analyzed them first in culture by electrophysiology. They had normal neurons, normal synapse numbers. This is why we did the electrophysiology. And we found, to our surprise, that when we measured AMPA receptor and NMDA receptor-mediated EPSCs separately, that the AMPA receptor EPSCs were changed, but the NMDA receptor EPSCs were not. This is surprising because if you have a change in neurotransmitter release presynaptically, you should have a similar change in both responses. The fact that this was selected for AMPA receptors indicated that this is a postsynaptic phenotype, whereas neurexins are supposed to be presynaptic. We confirmed that there were fewer AMPA receptors in the synapse, suggesting that this SS4 plus variant compared to an unrelated control, or the SS4 minus variant, has a decrease in AMPA receptors with a normal content of NMDA receptors, and inhibitory synapses were normal in these hippocampal cultured neurons. So these experiments raised two questions at this point. One question was, given that neurexins are presynaptic, but this is a postsynaptic phenotype, are we really looking at a postsynaptic molecule, a presynaptic molecule? Is this possible that neurexins are actually postsynaptic and that, you know, the presynaptic assignment was wrong, which can be very easily done because it's really not possible without functional experiments to differentiate between pre versus post. And the other question that these experiments raised is that since they were done in culture, does this actually happen in a real animal? Does this happen in a normal physiological environment? So to address this question, we used a preparation that we have used a lot in various experiments, whereby we inject presynaptically, in vivo, stereotactically, into the hippocampus, CA1 region, AAVs, which are viruses that allow us to express stuff. And in this case, we expressed Cre or delta Cre as a control in these presynaptic CA1 regions, neurons, and they are tagged with EGFP, so they're green. Then a couple of weeks later, we cut slices, and we record in these slices from subiculum neurons that were not infected with the viruses, as shown here, using stimulation of the axons emanating from the CA1 region and forming synapses on the subiculum. And with this preparation, Jason Aoto, who started these experiments more than 10 years ago, was able to specifically analyze two questions. One is, are neurexins really acting presynaptically because we have only a presynaptic manipulation? And second, does this presynaptic manipulation change the postsynaptic receptor responses? And to everybody's delight, Jason at that point, as shown here, found that the SS4+, the control here, suppresses AMPA receptor-mediated responses just as in culture, and that the presynaptic excision converting SS4+ into SS4- of neurexin 3 reverses this phenotype. So this was the basis for many, many additional experiments that I'm going to tell you about, demonstrating, I think, quite unequivocally that the presynaptic alternative splicing of neurexin 3 in this particular synapse, and only in this particular synapse. If I had time, I would tell you about other synapses where this doesn't apply. Okay? And I can tell you why this is. Depends on the ligands of the neurexins. That the presynaptic alternative splicing in this particular synapse dictates the postsynaptic AMPA receptor response. We generated and analyzed similar mice, also for neurexin 1 and neurexin 2. When we analyzed the neurexin 1 mice, we were surprised to find that there was no effect of presynaptic alternative splicing of neurexin 1 at SS4 on AMPA receptor responses. They were flat. They were just perfectly normal. But now the postsynaptic NMDA receptor response was changed. So the two different neurexins that are highly homologous have different functions. And you can see that the direction of the change was different. Now SS4+ causes an increase in the response, not a decrease. That could be reversed by the presynaptic excision of the SS4 exon in neurexin 1. Neurexin 2 manipulations conversely had no effect on either AMPA or NMDA receptor responses, suggesting that neurexin 2 does something different. So we have three neurexins. They have the same alternative splicing, highly homologous. The alternatively spliced exon is very, very similar. Nevertheless, completely different effects biologically. And they're all co-expressed in the same neurons, although to varying degrees. So these experiments demonstrated that the presynaptic SS4 alternative splicing on neurexin 1 drives NMDA receptor inclusion, increases NMDA receptor responses when there's SS4+. SS4- is inert, and if you overexpress SS4+, you have the same effect. Whereas the presynaptic alternative splicing on neurexin 3 suppresses AMPA receptors. This provides a mechanism of transsynaptic receptor control. It's a way how the presynaptic neuron can tell the postsynaptic neuron how many receptors to put into a synapse. And by using very economically the same class of molecules to do that for different receptors, you amplify the information content of these various genes that do this. How does this work? Let me go back and remind you of the interaction network that is mediated by neurexins and that I showed you very schematically and in abbreviated form before. What this slide shows is there's multiple interactions that are regulated by splice site 4, as shown here. There's also an interaction of splice site 2, but I'm not going to discuss it. This splice site 4 regulates the binding to LRTMs, neuroligands, and to cerebellins. But only the cerebellin interactions are mediated exclusively by SS4+ and not SS4-, suggesting that they are the best candidates. Now, cerebellins are secreted molecules, shown here schematically. They come in 4 genes. 3 is not expressed anywhere else but in the cerebellum. It is in non-functional anyway. 1 and 2 bind to one class of receptors called GLUDs that are homologous to AMPA and NMDA receptors but are not glutamate receptors. Cerebellin 4 binds to a completely different class of postsynaptic receptors called neogenin or DCC, deleted in colon cancer. The question thus arose, which of these cerebellins that are the only ligands we know that bind exclusively to SS4 plus but not to SS4 minus neoexons might be involved. We performed in situ hybridization analyses and single-cell RNA-seq analyses in the hippocampus to figure out where, if at all, cerebellins are expressed and which isoforms. And it turns out that cerebellin 2 is expressed in the subiculum, but not in CA1 region neurons. In CA1 region neurons, only cerebellin 4 is expressed in a subset of interneurons, not cerebellin 1, not cerebellin 2, at significant levels. You can always find some expression of any molecule in any cell. So this suggested to us that the cerebellin 2 is the best candidate, and therefore we generated conditional cerebellin 2 knockout mice and analyzed the effect of the cerebellin 2 deletion in the subiculum on synapses. Cerebellins are thought in the textbooks to mediate synapse formation, but we found that the deletion of the cerebellin had no effect on synapse numbers, as shown here. However, when we measured synaptic transmission, the cerebellin 2 deletion caused an increase in AMPA receptor responses and a decrease in NMDA receptor responses. This is the exact mirror image of the SS4 manipulations, consistent with the idea that the cerebellin 2 deletion disinhibits a suppressing neurexin 3 signal, and it reverses an activating neurexin 1 signal. Do these changes occur in the same synapses? We used minimal stimulation to test this. I'm not going to go into this. It goes a little bit too much into technical details. The experiment suggests that indeed they occur in the same synapses. So how do cerebellin binding, how does cerebellin binding to neurexins 1 and 3 actually do this? How does this work? It turns out, as I mentioned already earlier, that cerebellin 1 and 2 bind exclusively to GLUDs, of which there's two, GLUD1 and GLUD2, whereas cerebellin 4 apparently doesn't bind to GLUD at all, but instead binds to DCC and neogenin. So GLUDs are the best candidates for the receptors of the cerebellin neurexin complexes. We tested this by deleting GLUD1 in the subiculum and in cultured neurons from the subiculum because GLUD2 was not significantly expressed. And we used another recently developed approach for this CRISPR. And we used acute CRISPR, an approach that we are increasingly using, where you don't need to make mice or anything. You basically infect neurons either in vivo or in culture with viruses that mediate CRISPR-induced mutations or deletions. And as you can see here, if you have the right guide RNA, this can be quite effective, shown here by immunoblotting. We again measured synapse numbers, no change, just like with cerebellin 2 deletion. Measured synaptic function. The same phenotype emerges as for the cerebellin 2 deletion, an increase in AMPA receptor responses, a decrease in NMDA receptor responses. And double mutants demonstrated that GLUD1 deletions, neurexin SS4 manipulations, and cerebellin 2 deletions occlude each other. So they're genetically in the same pathway. So what I've shown you thus is that presynaptic neurexin 1 and 3 regulates postsynaptic NMDA and AMPA receptors via an intermediate that is identical for both neurexin 1 and 3, even though neurexin 1 and 3 have differential effects. This pathway is restricted to those neurons and synapses where there is a cerebellum, 1 or 2, and where there is a GLU-D, which is not true for many synapses in the brain. For example, in the hippocampal CA1 region neurons, we do not find any cerebellum, 1 or 2, or very, very little. And this pathway does not operate in those neurons. It doesn't exist in those neurons. In other words, this regulatory pathway can be specified by the expression of the ligands and splice variants. So how does the cerebellum 2 GLU-D1 complex transduce distinct to neurexin 1 versus neurexin 3 signals? The idea we had was that the only way this could possibly work is if GLU-D separately regulated AMPA and NMDA receptors via distinct cytoplasmic sequences. So we analyzed the cytoplasmic sequences that are conserved in GLU-D1 and GLU-D2. We identified 5 sequences that were highly conserved, and we performed rescue experiments with mutations in these 5 sequences that are shown here, in which all of the 5 sequences are replaced by the same mutant sequence to make them comparable, except for the C-terminal PDZ domain binding motif. We then performed rescue experiments, measured AMPA receptor responses and NMDA receptor responses. What you see here is that's the typical effect of the GLU-D1 mutant, an increase in AMPA receptor responses. It was rescued by all mutants except for one, mutant number 4. NMDA receptor responses, typical decrease. It was rescued by all mutants except mutant number 3. So different mutations impair the GLU-D1 dependent regulation of AMPA and NMDA receptors, confirming that GLU-D1 operates by bifurcating signaling pathways. We don't actually know to this date what binds to these sequences, but we believe that it's plausible to hypothesize that there must be specific binding proteins that then mediate this regulation that I just told you about. So what I've told you in essence is that 2 different neurexins that are highly homologous, regulated identically by alternative splicing, use the same signal transduction device to upregulate either NMDA receptors or downregulate AMPA receptors in synapses in which these signaling device components are present. I mentioned already, as indicated here schematically, that NMDA receptors and AMPA receptors are not only similar to each other, but also to GLU-D1s and GLU-D2. They are all one gene family. As you know, AMPA and NMDA receptors operate by opening a channel as a function of ligand binding. This channel is formed by multiple transmembrane regions in these receptors. So we asked whether the GLU-D operates by a similar mechanism, where you have multiple transmembrane regions that are present and that perform a conformational change that is then transduced into a cytoplasmic signal. To address this question, we simply replaced all transmembrane regions in the GLU-D1 or GLU-D2 molecule with a single transmembrane region from CD4, which is a lymphocyte surface receptor, totally inert, just the transmembrane region. We also deleted the ligand binding site that is inactive in GLU-D1 but binds glutamate and AMPA and NMDA receptors, and kept only the cerebellum binding domain and the cytoplasmic sequence. This is a very simple device. And when we used rescue experiments, we found, as shown here, that not only GluD2 rescued as well as GluD1, showing that at least these two molecules are isofunctional, but that these minimal molecules were also capable of rescuing the phenotype, both for NMAMPA receptors and for NMDA receptors, demonstrating that despite the homology between AMPA and NMDA receptors and GluDs, their mechanism is different. We then went on to specifically show that the same sequences that we identified with the mutations that I demonstrated to you earlier in GluD1 are actually capable of mediating the signaling. But I'm going to go over this because I think you get the point that I'm trying to make, which is that these experiments reveal one particular mechanism by which a presynaptic neuron can tell a postsynaptic neuron via neurexin signaling how many AMPA and NMDA receptors to insert. And I would like to emphasize or make you aware of how actually economical this is. Evolutionarily, then, you have a conserved family of proteins, neurexins, that in mammals at least acquire a site of alternative splicing that does a lot of things, including regulating their binding to a ligand to GluD. And GluD in itself, as our experiments show, among others, regulates AMPA and NMDA receptors, which are evolutionarily related to GluD. It's a very simple and economical way of solving a complicated question. You might ask how cerebellin binding actually regulates the cytoplasmic action of GluD to control AMPA and NMDA receptors, and I don't know how. I can only speculate that the differential affinities of neurexin 1 and neurexin 3 for cerebellin 2 plays a major role here in dictating the multimerization state of these molecules, because each GluD has two binding sites, and cerebellins in turn are bifurcated molecules as indicated here, so they would cause multimers. Multimerization of receptors is a common mechanism in biology for signaling, but we have not proven that this is actually what takes place. I would like to skip over the next sections because dinner is waiting, as we have already said before, and instead I wanted to close by presenting you a model. The model is that neurexins are signaling platforms. They are multimeric molecules composed of individually folded domains that bind to a vast number of transsynaptic and actually also cissynaptic ligands. And the way I would like to illustrate this is with the Swiss Army knife. This is supposed to be a neurexin. You have all these domains sticking out that are regulated by alternative splicing. So you could imagine that alternative splicing is like putting them in and out of the knife. And each one of these does something different. It's like a mosaic. It's a toolkit. It's a platform, a signaling platform. Which function operates at any given synapse? In this idea will depend on what ligands are actually present. And if multiple ligands are present, whether they compete for binding with each other and have differential affinities, which they always do. So one wins, the other one loses. Okay? Moreover, to make things even more complicated, some of the ligands at least, and some of the alternative splicing, is activity dependent. So it can change as a function of the neuron's activity and thereby change the properties of the synapses in an activity dependent manner. A form of plasticity that's obviously very different from classical NMDA receptor dependent LTP or LTD, because it regulates the number of receptors by a different mechanism. With this, I'd like to close. I would like to acknowledge Jinya Dai, who continued the work, the initial findings that Jason Aoto made 10 years ago. He's now associate professor at the University of Colorado. Jinya is now an assistant professor in New York. Their work was fantastically supported by Justin Trotter. I haven't really talked about the other people's contributions, and I'd like to acknowledge Xiaowei Zhang's wonderful collaboration. I'd be happy to take any questions, and I apologize that some of the stuff I discussed with you here is rather technical and maybe of limited interest, but I hope to have at least given you some taste, some feeling for how we are trying to approach the question of how wiring in the nervous system, how synaptic connections are actually made and specified. Thank you very much for your attention.
Thank you very much indeed, this overview about the synaptic technical, which is definitely gives plenty of new area to go for the future, and it still shows that how difficult it is to understand the function of the brain. So is there any question from the floor, from the students, or from the mentors?
Thank you. So you were talking about synaptic vesicles. I have a question about that. How do the synaptic vesicles find their way to the membrane, and how do they know what neurotransmitter to release?
Yeah, I haven't talked about that at all. I'd be happy to talk about that. That is work that we did some time ago. I actually published my last paper, I think, on neurotransmitter release mechanisms a couple of years ago, but we haven't really worked on this. The bottom line is that there are several stages in the molecular recruitment of synaptic vesicles to the site of exocytosis, which is referred to as the active zone at a synapse. These stages can be termed the recruitment, the tethering of the vesicles in a distant from the membrane, and the docking close to the membrane prior to fusion. And they are mediated by presynaptic scaffold complexes that we identified some years ago and that are composed of multi-domain intracellular proteins called RIM, RIMBP, MUNC13, and others. But these three are probably the central elements of this scaffolding machinery. And they basically capture vesicles first by a RAP-dependent mechanism, RAP3 and RAP27 in the case of the synapse. Then these vesicles get closer to the membrane and the snare complexes engage during the docking process to prime the vesicles for the calcium signal, which then triggers the snare and MUNC18 dependent fusion of the vesicles. So that's sort of the fundamental mechanism that emerged from this work. It turns out that that mechanism is universal in that many other fusion processes have a similar mechanism, except that the sophisticated tethering and docking by a scaffolding protein is unique to synapses and endows synapses with this incredible speed that is not observed, for example, for endocrine or exocrine secretion. But in principle, the mechanism of synaptotagmin-dependent calcium triggering of fusion that is mediated by SNERs and MUNC18s is very similar in all regulated exocytosis processes that I know of. Does that sort of...
Any other questions?
This was fascinatingly complicated. Why do you think the disease association is so non-specific of the deletions?
Because the diseases are non-specific. So in psychiatry, there's a huge problem of nosology in the sense that everything is based on how patients present, the natural history of the patient's presentations. And the psychiatrists can't really tell how many different schizophrenias or autisms there are, whether they are more similar to each other or whether specific subtypes are more similar to other subtypes of other neuropsychiatric diseases. I think the problem lies in the fact that the brain is so insanely plastic, and that depending on the experience of the individual, on the environment, and maybe also on other genetic background effects, patients react plastically to the same change in a different way. Remember that what you see in psychiatric disorders is not an overall dysfunction of the brain. What happens is that there is a dysfunction of a very selected subset of activities. So in schizophrenia, for example, which is really a truly tragic disease because these patients really suffer. These patients are tremendously burdened by their disease. A lot of the normal brain functions are still there. The patients have normal language functions. They suffer from increasing cognitive deficits, some of which may be due to the drug treatments, some of it are probably part of the disease. They have positive symptoms such as hallucinations, paranoia, and so on, but there's a lot that works just fine. Motor functions work just fine. Okay? So what I'm trying to say is that in neuropsychiatric disorders, the impairment of the patients is not a general impairment, like, for example, in Alzheimer's disease, where you have progressive destruction of the brain that eventually affects the entire brain. In schizophrenia, you have a very selected dysfunction, suggesting that the brain, in the case of neuroexon mutations, responds to the functional impairments that are caused by the mutation by plastic compensation processes that are differentially able to compensate, but can't compensate enough and not for long enough times. And individuals differ in how they react. Yeah. And some of it may very well be environmental. I don't think it's all genetic. I think it's a mixture, for sure. Yeah.
Thank you. I think we still have a time for a question.
Thank you for your presentation. My question would be that, is it possible to change the molecule of CNBLN2, or is it too specific that it cannot be changed in order to manipulate what information we transmit to the post-synaptic synapses.
Yeah, so could we develop, if I understand correctly, could we develop a drug that interferes with the system?
Yes.
Yeah. I think potentially, yeah. Absolutely. It's a signaling molecule, and there's no reason why we shouldn't be able to do that. One would have to have a good rationale to do that. So, for example, I could imagine that it might be a good idea to pursue this as a potential strategy for cognitive impairments in schizophrenia where there is tremendous evidence for an NMDA receptor dysfunction, although some people doubt that. That is by no means an established fact. Nevertheless, there is good evidence, especially on interneurons, and that may be possible to do that. Yeah. We have thought about lines like that. It would, however, only work in those synapses that actually normally have at least GLUDs. Moreover, what would make me hesitate just a little bit is that there is no evidence that GLUDs or cerebellums are actually ever genetically implicated in neuropsychiatric disorders, as opposed to neoexins, neuroligands, LRTMs, you know, the other ligands that I haven't talked about. So that, if I was a drug developer, which I'm not, that would make me sort of hesitate a bit.
Thank you very much.
Thank you very much. And again, thank you so much for coming here. I think it's still we can continue during the dinner, the discussion. And now I would like to ask András Varró to hand over the certificate and appreciation of your visit to Szeged.